In this video, we will discuss about an important pathway called as photorespiration. As the name is indicating, photo mean light, respiration mean intake of oxygen and release of carbon dioxide. But when this is occurring in presence of light, this is called as photorespiration. This is different from dark respiration. Now, we can define photorespiration. It is the light dependent uptake of oxygen and release of carbon dioxide by the green photosynthetic organs of a plant. So this occurs in the green plant and this occurs in the photosynthetic organs of a plant. This is also called as C2 glycolate cycle or PCO cycle. Full form is photosynthetic carbon oxidative cycle. This was proposed by Dacker and Teo in 1959. Now, during photorespiration, enzyme Rubisco, whose full form is ribulose 1,5-bisphosphate carboxylase oxygenase. In this photorespiration, this part, ribulose 1,5-bisphosphate oxygenase part will participate. Now, if photosynthesis is occurring, ribulose 1,5-bisphosphate carboxylase part will be activated. But here this part, that is ribulose 1,5-bisphosphate oxygenase is activated. So during photorespiration, this part of the enzyme Rubisco is acting, and conditions where this oxygenase part is favored, when oxygen is high, temperature is high, water stress conditions are there, loss of energy is taking place. So this is leading to photorespiration, right? This is loss of fixed carbon dioxide. But in case of when photosynthesis has to occur, then carbon dioxide concentration will be high, temperature is low, optimum water, optimum temperature, water stress conditions are not there, proper availability of water is there, then C3 cycle or Canvin cycle will be operative and enzyme part will be activated is ribulose 1,5-bisphosphate carboxylase. And here which part is activated? Ribulose 1,5-biphosphate oxygenase plays an important role in the photorespiration. This we will remember, which part of the enzyme is active in the photorespiration. Now, during Calvin cycle, there is formation of sugar, but in case of photorespiration, there is a loss of photosynthetic products. So, this is different from the normal respiration. Now, cell organelles which are involved in photorespiration are chloroplast, peroxisomes and mitochondria. So, three cell organelles plays an important role: chloroplast, peroxisomes and mitochondria. Environmental factors which favor the photorespiration, they are when temperature is high and oxygen concentration is high, water stress condition or drought condition mean when there is less availability of water. C2 cycle is called because there is synthesis of two carbon compounds. So, this is called as C2 cycle. C is carbon. So, we'll discuss three cell organelles where it is participating, like this is occurring in chloroplast, peroxisomes and mitochondria. Now, we'll discuss in detail how these reactions are taking place. First, we'll discuss about chloroplast, right? So, this is the reaction where in chloroplast is taking place. I'll just enlarge it just to make you understand. Right. This is the chloroplast, right, where these reactions are taking place. So, here ribulose 1,5-bisphosphate, this is the first substrate, CH2OP, C double bond O, HCOH, HCOH, CH2OP. So, at first position and fifth position there is phosphate group. Now, this will be acted by enzyme ribulose 1,5-bisphosphate oxygenase because oxygen concentration is high. A water stress condition may be there. So, this part of the Rubisco will be activated and this ribulose 1,5-bisphosphate in presence of this enzyme. Now, this will break up into phosphoglyceric acid and phosphoglycolate. Now, this is 2 carbon in length and this is 3 carbon in length, right? So, this is phosphoglyceric acid COH, CHOH and CH2OP and CH2OP and COH, this is called as phosphoglycolate. Now, this phosphoglycolate will enter into the photorespiration. Now, this will enter into the C3 cycle. This will go into the Calvin cycle. In detail, Calvin cycle I have discussed in separate video and link will be shared in the description box. Now, we'll see here what happens to phosphoglycolate. Now, this phosphoglycolate will lose its phosphorus, right? This will be accepted by water, and water will be converted to phosphoric acid H3PO4. So this loss of phosphorus is called as dephosphorylation. Now this will be accepted by water, and when this loses phosphorus, then this phosphoglycolate will get converted to glycolate. Now when this has been converted to glycolate, now this will enter into another cell organelle that is called as mesophyll tissue, that is in the peroxisomes, right? Now in the peroxisomes, this glycolate will be oxidized. This is called as oxidation in presence of oxygen. Now this will be forming a side product. This is called as hydrogen peroxide. Now this hydrogen peroxide will be catalyzed by catalase enzyme, and this will be converted to water and oxygen. If hydrogen peroxide as such accumulate, this may become toxic. So this will be acted by catalase, and this will be converted to water and oxygen. Now this glycolate, after oxidation, this will form glyoxylate. Now this here will be converted to aldehyde group, CHO group. So this is called as glyoxylate. Now this glyoxylate will accept one amino group at this position, right? This will accept one amino group, and this will be converted to glycine. Now amino group will be transferred to this glyoxylate by enzyme transaminase. And from where this amino group is coming to this glyoxylate, that is from amino acid. Glutamic acid, COH, CHNH2, CH2 twice, and COH. This is called as glutamic acid. So this will lose its amino group to this, and this will get converted to glycine. And when this has donated its amino group, now this will be converted to alpha ketoglutaric acid. At this position, keto group is created, COH, keto group, CH2 twice, and COH. This is also called as alpha ketoglutaric acid. And when this alpha ketoglutaric acid accept amino group, now this will be converted to glutamic acid. When this loses amino group, this get converted to alpha ketoglutaric acid. So here glyoxylate accept this amino group and get converted to glycine. Now this glycine will be transported into another cell organelles. This is called as mitochondria, because at the membrane transporters are there. So that will help in transportation of the glycine toward the mitochondria. So here in the mitochondria, two molecules of glycine, they will condense, right? So this is one molecule, this is another molecule. When they condense with each other, there is loss of carbon dioxide, right? So there is also loss of one amino group. Now they will be lost. Now when they are lost, amino group is lost, deamination, carbon dioxide is lost, decarboxylation. So this is being catalyzed by enzyme glycine decarboxylase complex. Now two molecules of glycine, and in presence of NAD, will get converted to NADH2, and this is again reduced, we can say NAD is getting converted to NADH2, and two molecules of glycine will get changed to one molecule of serine. There is one amino group, because in two molecules of glycine there are two amino group. So there is loss of one amino group, one carbon dioxide. NAD is getting converted to NADH2, and this will be getting converted to serine. Now when in mitochondria two molecules of glycine have been converted to serine and there is loss of carbon dioxide, like we define photorespiration, that this is intake of oxygen and release of carbon dioxide. So we can see here there is loss of carbon dioxide. Now this serine will enter into this will enter into peroxisomes, right? Now this serine, when enters into peroxisome, this will lose its amino group, and this will be accepted by alpha ketoglutaric acid, and this will get converted to glutamic acid. And after losing this amino group, serine will get converted to hydroxypyruvate. So at this position there is conversion of this group, amino group, into keto group. So CH2OH, keto group, COH, this is called as hydroxypyruvate. Now hydroxypyruvate will be reduced in presence of enzyme reductase, and NADH will donate its hydrogen, get converted to NAD, and this hydroxypyruvate at this position will accept these two hydrogen. So this double bond will be dissolved. This will accept hydrogen here. This will be single bond. Now this will be converted to glycerate molecule. So this hydroxypyruvate have been converted to glycerate molecule after reduction, after getting two hydrogens. So in peroxisomes, what reactions are taking place? Glycolate is being oxidized. This is getting converted to glyoxylate. After accepting amino group, this get converted to glycine, and glycine will enter into the mitochondria. Two molecules of glycine will condense, loss of amino group, loss of carbon dioxide, and this will get converted to, this will condense and form serine. This is again an amino acid. Glycine is also an amino acid. So this serine will lose its amino group when it enters into the peroxisome, and this will get converted to hydroxypyruvate, and hydroxypyruvate is getting converted to glycerate. Now glycerate will again move into the chloroplast. So in chloroplast, glycerate will be taking phosphorus from the ATP. What is ATP? Adenosine triphosphate. So it has three phosphate. This will get converted to ADP, and this phosphate will be accepted by this H group in the presence of enzyme kinase, and this will get changed to 3-phosphoglycerate. Now this 3-phosphoglycerate will get changed to ribulose 1,5-bisphosphate. So this cycle will be completed, and ultimately again this can be acted by enzyme Rubisco, that is ribulose 1,5-bisphosphate oxygenase, and this will form phosphoglyceric acid and phosphoglycolate. So this again I'll repeat, ribulose 1,5-biphosphate in presence of oxygen, high concentration of oxygen, and ribulose 1,5-bisphosphate oxygenase get converted to phosphoglyceric acid and phosphoglycolate, and phosphoglycolate further enters into the chloroplast, then peroxisomes, then into the mitochondria, and again glycerate is being transported back to the chloroplast, and this is getting converted to 3-phosphoglycerate. Now 3-phosphoglycerate will again react with these molecules and get changed to ribulose 1,5-bisphosphate, and in this way this cycle is completed. So there is intake of oxygen, if we see, and there is release of carbon dioxide. So this is called as intake of oxygen, release of carbon dioxide, respiration, and this is occurring in photosynthetic organs, and in eliminated green cell this is called as photorespiration. So this is all about cycle of photorespiration, and if I shorten it, and you can see here, this will be appearing like this: chloroplast, peroxisomes, and mitochondria. So this is a formation of C2 compounds in this cycle. That's why it is called as C2 cycle. Now, what is the importance of photorespiration? Because here no ATP is generated, so photorespiration does not produce energy, rather this consume energy. During photosynthesis, there is formation of sugars, but in photorespiration, there is loss of sugars or loss of fixed carbon dioxide. So because during photosynthesis, when more nutrients are produced, more carbohydrates are produced, more yield is there, more biomass is produced, but in case of photorespiration, there is 25% loss of fixed carbon dioxide, leading to loss of biomass, leading to loss of yield, because this is not producing any energy. This is a highly wasteful process. This helps in dissipation of energy when stomata are closed during water stress condition during daytime, because during daytime stomata close when there is drought or less water availability. To reduce the rate of transpiration, stomata will close up, so this will help in dissipation of the energy. When intense light is there or temperature is high, so photorespiration may prevent the chlorophylls from photooxidative damage, because chlorophylls, they are an important part of photosynthetic process, and if chlorophylls are damaged, no photosynthesis will occur. So photorespiration may prevent photooxidative damage of the chlorophylls by dissipating excess of excitation energy, and this also helps in maintaining the redox potential in the cells or redox balance in the cells. So this will help in maintaining the redox balance, and this also support plant immune defense responses when some pathogen attacks the plant, right? So a plant try to fight with that pathogen, and that is called as defense process. So photorespiration may help plant supporting in immune defense responses. So this is all about photorespiration. If you have any question, you can ask me in the comment box. Thank you for watching my video.